now returning to the University of Pittsburgh, as our next speaker is Mauricio Rojas, who will talk about the mesenchymal stem cells lose regulatory functions in patients with pulmonary fibrosis, idiopathic pulmonary fibrosis. Thank you.
Thank you very much. And okay, Lucas leaving, but I wanted to thank him for the opportunity to be here, and not only for that, to give me the excuse to be in Italy for the last week driving around this beautiful country. And okay, I'm going to talk about a little story that we've been working in the lab about aging and why we believe aging is an important component, not only for pulmonary fibrosis, but basically for the large majority of the lung disease. We have here that we all know that acute lung injury, the incidence increase with age. We know that the susceptibility to lung pulmonary infection, and we just learned about COPD, emphysema, how all of them increase with age. And I have here this nice picture because I like to remind all of us that aging is not that our fibroblast age. It's not that our epithelial cell age. It's a universal process. The whole body, we have defects at every single organ. We all know that bone marrow, you have a fracture in the bone when you are 70. It's going to be totally different if you have a fracture when you are 14. And it's something that we need to remind all the time. Remember that. And here it's important to say that basically what is common here is we have defects in the mechanism of repair. Why is that? And it's what we've been very involved in the lab, trying to define what are these mechanisms of repair that are affected with age. Here, because we are in a pulmonary fibrosis, we are going to focus today on IPF. And you all know this slide, and we all know these incidents. And basically, if you look, 55 is a cutting point. After 55, you have 85% of the cases of pulmonary fibrosis. And this is important too for one of the discussions that we have been having here during the meeting. It's animal models. If you think that a mouse, a 1-year-old mouse, is an equivalent to a 40-year-old human, basically when you go and treat with bleomycin and you try any therapy in an animal that is 3 or 4 months, you're basically in this range, in a 0.8%, where basically we don't have IPF. And that can be one of the many explanations why we fail in all the therapies and the translation of all the very nice observations that we had in the bench when we go into clinic. To study pulmonary fibrosis and aging, we started several years ago, and we treat 2-year-old man. Was difficult to do it. Took a lot of training. and basically, we treat animals, 2-year-old mice, with bleomycin. We gave intratracheal. I mean, that was the traditional way at that time. And basically, what we observed was when you treat a young animal, 14 days later, we have a severe but not little pulmonary fibrosis. But when you treat an old, basically a 2-year-old mouse, the fibrosis is basically in the whole lung, and what is quite interesting, it's progressive. We look animals 3 or 4 months later, and the fibrosis was there. And that was basically reproducing what Victor has shown us a couple of hours ago. We did quantifications, collagen deposition in all of these animals, and it was clear that all animals have more collagen in their lungs after injury. Then Anna Mora, she developed a mouse model of gamma herpes virus infection. Basically, she took, at that time, a susceptible host. She was working at that time with gamma herpes virus and the interferon gamma receptor knockout animals. These animals, young animals, 3 or 4 months animals, when they were infected with gamma herpes virus, they developed pulmonary fibrosis. But when she was using wild-type animals, these animals didn't develop fibrosis. She needed to produce these mutations in the receptor of the interferon gamma. Oops, I think I pushed the wrong button. I think I did. I think I killed the presentation. Okay, now we are going into a black box that we don't understand. Okay.
So push it again.
Push it again. Nothing happened. Okay, great. Here it is. Thank you, Paul. You know, you're a man of many talents. Okay, but what she did later is she took all animals, all C57. In this case, they were wild type. And she infected with gamma herpes virus. And then she compared with young animals infected with gamma herpes virus. And basically what we have here, the young animals infected with gamma herpes virus 15 days and 250 days later. Basically the animals recovered. Like the previous experiments, those animals didn't have any consequences as the infection. But here she took all animals. Remember, these were wild type. There were not any defect in the interferon gamma receptor. But when she looked 15 days and even 250 days later, there was a severe progressive pulmonary fibrosis. And we have here 250 days. And this is important for two reasons. One is, okay, the susceptibility, and the second, it's we traditionally think that IPF is the result of continuous and multiple injuries across the life of the individual. And this data can suggest that it is true, but what is important is the moment that you have, the moment in your life when you have your injury. I mean, here the animal was old, all these mechanisms of repair were affected, and when this animal got injured by the infection with gamma herpes virus, basically they couldn't repair. And the only mechanism that the organism has, I mean, or you die or you try to hold the dam and the levee, otherwise everything will flow out, is scar tissue, and that's what basically they do. And this was not a defect in the immune response. Basically, she looked for the clearance of the virus, and she looked in young animals and all animals. Basically, by day 7, they have the same title of virus. Here in black dots are the old animals and the white, and this is the number of copies of viral DNA, and basically there were no difference. I mean, there was it's not a defect in the clearance of the virus. It's a defect in the mechanism of lung repair. If we talk about lung repair and what Luis had extensively explained about mesenchymal stem cells, and I think it's one of our babies that we've been working for a long, long time. And this is a very complex mechanism. There are so many different paths in which a mesenchymal stem cell can prevent and be involved in the process of repair of the lung injury, any type. I mean, we go here. It's what Luis already had explained: the microparticles and the mitochondrial transfer. It's always important to remember these cells. They go into the lung. They will participate of the repair. But if you look 14 days later, like in this experiment where we treat animals with LPS, we infuse the animals with GFP positive cells. You can see clearly 24 hours later, but basically 14 days, at the 14 days later, all the cells are gone. It's a very transient effect in these cells, by these cells. We did, or if we follow what Luis had published basically in '93, where he treated animals with bleomycin, and we infused the animals with mesenchymal stem cells and was able to demonstrate protection. What we did, we took a next step, and what we did depletion of the bone marrow. Basically, we gave busulfan to these animals to deplete the bone marrow, and we found that if you deplete the bone marrow of these animals and you gave them bleomycin, the animals die. But only by giving mesenchymal stem cells systemically, these animals recover, and we don't have mortality. And it's clearly, we have a decrease in the severity of the injury. Here is the animals treated with busulfan and bleomycin. Basically, they don't have bone marrow. But when you gave stem cells, it's clear there's a decrease in the severity of the injury induced by bleomycin. But what we know about mesenchymal stem cells and aging? other people has been interested in the field too since 2008. It's been published that they look different, and they proliferate less. And what is interesting, they express markers of senescence. To try to test that, we developed in the lab a protocol in which we try to isolate these all mesenchymal stem cells. And basically what we do a complicated sorting using some of the markers that Luis already expressed. And we isolate fresh stem cells for the different studies. Our first observation was in these animals, all stem cells have a decrease in the expression of several markers. During the meeting, you have seen these yellow and purple heat dots, and it's clearly that purple, what is downregulation of the expression of some genes, it's clear that these all stem cells decrease the expression of several of these genes. And what is quite interesting is the genes that are affected by age are genes that express proteins that are involved in migration of the cells and cell recruitment. To study that, we did simple migration assays, and where we confirmed that all cells don't respond equally to factors that are known to be involved in cell migration, like SDF, IL-17, and 6CL3. But to go into the biology, we did parabiotics. You all know how the parabiotics works. Basically, you tie a GFP animal to a non-GFP. By 14 days, these animals are going to share circulation. Here you have as a blood sample from a wild-type animal, and 50% of the blood basically is shared by the GFP positive, meaning they share circulation. When we did migration assays, I'm going to go here straight. First we did different combination of animals. We did old versus young, and here we have first old, and you believe, I don't know, it looks a little bit older. And you induce injury to the wild type, old wild type, and you see how much old MSCs goes into the lung? Basically none. So cells are not recruited from the young one to the old one. Opposite happens when you are a young animal and you recruit young cells. That's a good number of cells. Mesenchymal stem cells are recruited into those lungs. But what is quite interesting comes here. You are a young animal, but you have all the stem cells. You can't recruit cells. Sorry, the opposite. You have an old lung and young stem cells. The cells can migrate. They will respond to the signal produced by the lung, and they're recruited into the lung. But when you take an young animal that is tied to an old animal, cells are not recruited, suggesting that basically you are old as your stem cells. So that if you are used or for some reason your pool of stem cells is defective, your ability to respond to injury is going to decrease. And this is associated and correlated with the severity of the injury. Here we hear about fibrocytes. That is, for some people believe that it's important in the pathogenesis of fibrosis. And here that doesn't happen. But it's quite interesting. Age doesn't affect the recruitment of fibrocytes. Only happen in mesenchymal stem cells. What is mesenchymal stem cells? At the same time, they have a decrease in the ability to differentiate. And I'm going to go quickly with these couple of slides because I want to go into the important point here. We isolate mesenchymal stem cells from all animals, and we gave 2 animals that we treated with LPS. And these all cells were less effective, reducing the pulmonary edema and the inflammation induced by LPS in those animals, suggesting that yes, all the stem cells, they have a defect in the ability to prevent injury. I was giving this talk to the support group that Kathy runs there at the Simmons Center, and a patient come and say, "Hey, doc, the only thing that I understood of your talk is that it looks like the cells that I have in my bone marrow are important for the etiology of pulmonary fibrosis. Why you don't go analyze my cells. And basically with Kathy, we had created a protocol that patients that go what is called warm autopsy. There's a group of patients that donate not only their lungs for research, but now they are donating their bone marrow. They gave us part of a vertebral body for us to study the characteristics of the stem cells on those patients to have IPF. And that's why I'm always very grateful, because this is a patient initiative. It's nothing that I came and I wasn't smart enough to ask for this, but these guys, they believe this can be an important part to develop the knowledge of what happened in IPF. And that's exactly what we do. We get vertebral bodies from patients that had died and had donated their vertebral bodies for the research. And using a protocol that was developed by Albert Donenberg at the Hillman Center in Pittsburgh, we isolate the mesenchymal stem cells from the bone marrows of from the vertebral bodies of these patients. Here is some of the markers and some of the characteristics of those cells that we isolate. And we look the traditional things in those cells, and we find that these cells, they have, this is the control. We use as a control donors of lung for lung transplants. At the same time that patient donates their lung for lung transplant, we get a vertebral body that we use as a control. And here we have one of those donors, and we look for telomerase. And the expression of the telomerase is clearly that these controls, by red, have a high expression. But in the patients with pulmonary fibrosis, basically the expression is not there or have decreased. We did some studies on activity. Clearly, the activity in the IPF patient is lower when you compare with the controls. These are MSCs obtained from patients with IPF. We look for mitochondria, and this is mitogen for mitochondria content. And we can clearly see that non-IPF have more mitochondria. When we did quantification, IPF MSCs have less mitochondria when you compare with the non-IPF. And here going with Victor's paradox, that it's something that we don't understand very well right now. When we did gene expression, here it's clearly that these are the blue dots correspond to the IPF patients. The red dots correspond to controls. They separate in different groups, clearly. But when you look at the gene expression, opposite to what happened in the old cells, and it's something that we are trying to study right now. IPF patients, the MSCs from IPF, they're upregulating several genes. And some of these genes are genes that are involved in development differentiation, and we don't know what exactly that means right now. It's part of the ongoing studies. But they have a downregulation of cell function and migration, and some of the genes or proteins that are involved in the protection, like IL-10 and HGF, are downregulated in these cells. And this is part of the ongoing work, and I think these are the nice data. I mean, when you get something that is totally opposite, they open a whole window for research, and we are quite excited. But functionally, we were very excited about this data until Serpil, she published a paper using bone marrow cells and pulmonary hypertension. She did the same, but we did for pulmonary fibrosis. We took animals, and we gave bleomycin, but at the day one, we injected with MSCs obtained from the patients, with IPF patients, and with normal controls. This is the weight. Basically, animals that were treated with MSCs obtained from IPF patients, they were less effective to prevent the decrease of the general state of the animal. But what's quite intriguing was the data of the histology. Here we have bleomycin, day 7, day 14, normal histology. Here we have MSCs from control patients, non-IPF patients. At day 7, they control inflammation as always, and decreasing the severity of the inflammation and the fibrosis by day 14. But MSCs obtained from IPF patients were not able to control inflammation at day 7, and the severity of the injury was more pronounced in these animals, suggesting that these cells were pro-fibrotic. They were increasing the fibrosis in those animals, different than what happened normally with MSC. Finally, as a conclusion, we believe that anything that can affect a stem cell can be DNA damage, we know. We were extremely happy to listen to Ivan when he told about metabolic syndrome, because we know that metabolic syndrome is one of the consequences of a stem cell exhaustion. And all of that is basically going to decrease the number of the cells, and it's going to decrease the ability of the cells to respond to injury. And basically, when you decrease your ability to repair, the only option that you have is to create a scar tissue, and that's what happened with fibrosis. Finally, I want to thank, this is the people, the translational group at PIT, the postdocs, Naira, Eder, Maria that was here, that she left. She's flying right now back to Pittsburgh. The Simmons Center, everybody, Naftali, and everybody, all the people at the Simmons Center. But what I think is more important, the patients that are the inspiration for our work. Thank you.
Thank you.
Maurizio, thank you for that great presentation. The concepts that you've developed for the concepts that you've developed for age-related changes in bone marrow-derived stem cells, do you think the same? I mean, as you know, there's a mesenchymal stem cell population that's been described within the lung, adult human lung. Do you think the same concepts apply, and have you looked at that at all?
That's a great question, and we believe the same happened, but unfortunately, I haven't looked yet. But it's clear, like I tried to say, it's a universal thing that happened in the bone marrow, in the lung, in your heart, in your brain, everywhere. Unfortunately, the clock is clicking every day, and we are getting old and old, and it's happened everywhere. It's not that your lung age faster than your, no, we all basically age at the whole body at the same pace.
So I don't know whether to be totally depressed and go out and buy a gun and shoot myself now, or cautiously optimistic, because I'm not sure in your control cells at the end, were they age-matched control cells, non-IPF?
That's a great question, and it's a 10-year difference, I would say, between the IPF and the NOR. And it's part of the studies that we are doing, because basically we need to compare, we have three things going on. One is age, and from age to disease.
Yeah.
And it's part of this triangle that we need to play. Now we are moving the age into the disease, but it's important. And there's the important thing about what you say, because a lot of people work about autologous stem cell transplant. Mm-hmm. And we don't believe that it's appropriate, because your stem cells are going to age too. And what you want is to have a source of very young stem cells. That's why I think these amniotic stem cells are really good, because they are 0 miles. Exactly. They're 0 miles, and they're more appropriate for any kind of intervention. If you go and use the fat tissue from a 70-year-old to treat IPF, that can be a problem. And, but you're absolutely right. We need to try to match the control closer. We have a 10-year difference between one group and the other.
Well, it begs the question, though, which comes first? Is the disease giving rise to an abnormal bone marrow response, because the disease tissue may be putting out mediators that modify how the bone marrow occurs, just as readily as the bone marrow giving rise to an aberrant healing response up in an injured lung? So I think you have to really have age-matched controls to
Yeah.
even raise that question.
Yeah, no, and we try to answer that with the mouse experiment or the mice experiment. And my bias right now, it's with age, we have a decrease in the population of mesenchymal stem cells at the bone marrow. We have a decrease in the ability of these stem cells to respond to injury. That's why when you have injury and you are 70, you are going to respond totally different than when you have the same type of injury and you are 14.
So I'm going to go get the gun.
Hey.
Yeah, so you showed that mesenchymal stem cells from young mice are recruited to the injured lungs of old mice and provide protection from death. But you said that the same thing doesn't, that young fibrocytes are not recruited. So I wasn't, you went by that quickly, so I wasn't
I'm
sure
sorry, yeah.
how you drew that conclusion. And also, is there any overlap between fibrocytes and mesenchymal stem cells?
Wow. First part, what we had found is old stem cells they don't respond to the signal of the lung, no matter if the lung is old or young. But when you have young stem cells, cells respond to the signals produced by an old and a young lung. That means old stem cells don't respond to signals. Young stem cells, yes. But it's not a defect in the signal produced by the lung. It's a defect in the cells to respond to that signal. And the second part of the question, that is quite interesting because we're talking about plasticity of the cells. We know that fibrocyte and mesenchymal stem cell, they look very similar, but it's a matter of expression of markers. We know that fibrocyte is basically a hematopoietic cell. It's a CD45 positive cell that expresses 6CR4 and produce collagen. By definition, mesenchymal stem cell, as Luis presented, is a CD45 negative cell. One of the main functions of mesenchymal stem cells or mechanisms used is modulation of the immune response. That has not been clearly demonstrated in fibrocytes. Some people argue that yes, they have some potential to modulate the immune response, but it's not that clear as has been previously demonstrated in mesenchymal stem cells. They are similar, but they are not the same.
Okay, thanks.
So years ago, we looked at some natural experiments that were relevant to this, and then I had teenagers and I ran out of all my own energy. But I'm asking you to maybe think about the natural experiment you could pursue these. So years ago, we looked at patients who had bone marrow transplants and had surgical lung biopsies for a reason, for a clinical reason, and showed what we thought was that stem cells could replace all the non-hematopoietic and hematopoietic cells in the lung. But one of the questions that I've asked our bone marrow transplant people, and they're not interested in thinking about this anymore, is whether you could take mismatched aged bone marrow transplants, i.e. older patients who get very young bone marrow transplants, and to see the relative incidence of lung injury, particularly obliterative bronchiolitis and other fibrosing lung diseases that might occur as a result of the conditioning regimen, including radiation and chemotherapy, since we see some of those patients who develop progressive fibrosing lung disease. So they tell me that they've looked at this, but nobody's ever published anything, and I think that there might be something there that might be relevant to what you're thinking about.
Yeah, I think it's a great idea. Thank you.
So something for the students in the audience to be thinking about is how do you deprogram this aging process, because it's a superimposition on developmental processes, and some of it's bits falling off and genes getting smashed with radiation and stuff like this, but there's also a program in there. And you know, IPS technology shows you that you can actually drive things backwards to a de-differentiated stem-like phenotype. And the question is whether you could do that effectively and efficiently in aged cells. Right now it's extremely inefficient and probably not scalable, but that's something that would be worth spending some time on, because that would actually be that would really be transformative if you could do that. So then you could do autologous transplants with a rejuvenated cell lineage, as opposed to going to non-autologous using fetal material, which is a great solution, but you still may have mismatch issues.
Yeah, no, that's a great idea. And please do it quick.
That's going to be a 40-year problem, by the way.
Exactly. Thank you.
Fantastic.
Thank you.